great pleasure to introduce Raquel Gore, who is a longtime friend and colleague, and really has just been a wonderful contributor to the field in many, many ways. She's incredibly accomplished, and I'll tell you a little bit about her background, but first I want to just say a little bit about her work and her research. She's really been a force in the field. Her work has mostly been related to schizophrenia, but also very importantly related to early development and all of the factors that come together from the standpoint of genes, environment, demographics that shape an individual's risk or resilience in relation to their development. She's been at the University of Pennsylvania for her entire faculty career, and there, with her husband and others, has established a research program that is internationally known. And in addition to the science that's come out of that, has been an incredible resource and continues to be an incredible resource for the field. And one of the things that she'll talk about, I'm sure, is the neurodevelopmental cohort that she has established. We were just talking about this morning, that has, I can't remember the exact numbers, 9,400 some kids that are no longer kids, that have developed and are in their 20s. This rich cohort has been longitudinally followed, phenotyped in many ways with cognitive and emotional and all kinds of other measures, imaging data, genetic data. And you can imagine what a treasure trove this has been, and thousands of papers have actually come out of this already. But in addition to that, Raquel was telling me this morning that they've also developed a cohort of the offspring of this sample. And in collaboration with the Children's Hospital of Pennsylvania, she's developed sort of an institute, as I understand it, that now encompasses not only the first generation, but the second generation of these children. And obviously, from the standpoint of thinking about risk related to schizophrenia and other disorders, this is really, really important. Raquel's work has also really focused on genetics from the standpoint of polygenic risk scores, but also rare genetic variants. And both of those are important in thinking about risk to develop psychiatric illnesses, and have done some very creative work actually looking at the combination of risk using some rare genetic variants like copy number variants and also polygenic risk scores. So really very creative strategies related to what she's been up to. She's been incredibly productive. I looked at her CV this morning. It's over 600 publications. So, I don't know when you're getting any sleep, Raquel, but it's quite impressive. And also has been a leader in our field. She's been the president of many of our societies that are the most important societies, the American College of Neuropharmacology, Biological Psychiatry, and others. She just won a very distinguished award from the International Congress of Neuropsychopharmacology, and many, many other awards. Her background is interesting and important to keep in mind as you hear what she's going to talk about, because she has a PhD in psychology. She has an MD, and she also did a residency in neurology and in psychiatry. So, incredibly well-trained to be in this position, but most importantly, has really dedicated herself to the field and to the patients that she's been struck by over the years. And it's really exciting for us to have you here, and particularly special from the standpoint of, having been friends and knowing you over so many years, to get you out here to be part of this. So thank you so much for coming, and let's give Raquel a really nice welcome.
So thank you for the nice introduction. And Ned and Richie have been friends for many, many years. And it is just a pleasure, really, to see you again and be here. And the contribution that you have made in Wisconsin to science and to translating it, from non-human work to human work to clinical population is really incredible and sustained over the years. So it's a true pleasure. So what I will do today is I'll try to cover 3 points. One, we know that when we are born, we are dealt with the genes are from our moms, are from our dads, and that's it. There's no control over it. You're dealt with a set of cards. The set of cards can explain, in most people, very small part of who they are going to be. So who we are going to be, each one of us, is really shaped by our environment. And a lot of effort in the past couple of decades has been devoted by the National Institute of Mental Health that supports research on mental illness, has been devoted to genetics because there's been tremendous progress in other fields of medicine. For example, cancer, in understanding more the biology of cancer, which is critical for innovative approaches for treatment. And when you see that in one area and you want to push forward, you think about what can we do in our area? And it's very complex. So NIMH has invested a lot in genetic studies, which is good. And many times the geneticist will say, environment is messy. Give me a number. What do you mean by environment? Environment is a dynamic process, but we now have tools to quantify it. So the challenge and some approaches that can be done to study the environment, and I saw it yesterday in the posters. Many of you are doing it already. So that's the next wave, the next generation that many of you, hopefully, will proceed with. We have the tools to advance the knowledge that relates to brain and behavior. The second I will highlight in more detail is the exposome. Another word for what we are exposed to, our environment, in common variant, the general population, and in rare variants that Dr. Kelly mentioned. And then give you some example of how can we move forward. There's no one way, it needs multiple of approaches. So the mission that I think have guided what I'd like to accomplish and what many of us would like to accomplish is to transform the clinical care of our patients. And this has to start at a very young age. When, I've seen thousands of people with schizophrenia, an extreme form of psychopathology, usually late teens, early adulthood. And over the years, I've lamented, if I would have met them earlier, would I have been able to help tweak, not I personally, but with collaboration with the families, with collaboration from other investigators, mental health professionals, to tweak the neurodevelopmental trajectories. So if the break occurs, they will be able to stand up and it will be a softer landing. They will have the capacity to continue with life and have some quality of life. And that's what drove me to go earlier on. So if you look at the blue circle, is the phenome. This is what people present with. And when we talk about phenomenology, we talk about the symptoms. People come with a chief complaint. Many times I tell colleagues who think, well, where are the patients coming from? We don't grab them off the street. They come to us because they are undergoing some distress and they need some professional help. So I'm talking clinically now. So they come with symptoms. They have behaviors that we are trying to link to brain function. So I call it neurobehavior, okay? In multiple domains, all coming from the brain. The brain produces phenotype in brain structure and brain function. And ultimately, it's the activity, the outcome, the level of functioning that we would like to be able to improve. And this is clearly continuous dynamic interactions with the genome, common variants affecting most of the population, and some misfortunes like mutations that can take place and impact behavior, and epigenetic factors from the get-go that modify our DNA. And then the exposome that is very rich, the environment. And when does it start? Intrauterine. That's the first environment where the fetus is developing. And there's going research in animal work and in human work showing the impact of intrauterine environment on the developing fetus. But then the process of inflammation, toxins that we are affected to, trauma, stress, all of this work together. And to accomplish this, we need an intergenerational approach that I will illustrate. So from the perspective of development, I look at it as steps, and that's a courtesy of Tony Lamentia, who is studying 22q11.2 deletion syndrome that I will illustrate today. It's a series of steps, and they can be small misfortunes or falling, tumbling early on. It's polygenic, it's not one gene, multiple genes impact it. It's a sequence that is highly structured, that goes from patterning, for example, to proliferation, to circuit formation, and takes years. Any tumbling early on, we need the ability to compensate and move on. The greater, if people get up, I mean the kids get up, they will have a what we call neurotypical development. The greater the stumble, more repeated across the step, the more we will have a deviation from normal. And we measure, you know, if you remember, or your siblings, or your children, the red or pinkish and bluish developmental curves for height, weight, head circumference. And when you come with a child to the pediatrician, they say, oh, they're right on the curve. Oh, they're 95% in weight, great, in height. We want to be able to have developmental curves for brain and behavior. Not to point out something is wrong with you, but to tweak the developmental trajectory as much as possible and identify children at risk and do something about it. All right, so when we look at GBAE, why is it so difficult? At times, you know, after spending all of my career, many years studying it, it feels like, yes, we're making progress, but we are not there yet. And we are not there yet, not because of lack of motivation or you know, critical mass of people who are interested, because it's complex. Think about it. We will never be able to implant a brain. The brain is who we are. You can take a kidney out, you can cut part of the liver out, you know, you can do cardiac, not brain. It's very, very complex. So there are multiple connected regions. It's enough that one region is impacted, one circuitry, and the impact is like a domino effect. And we are studying it now. There were no, I mean, the science, the neuroscience looking at networks. We didn't have the ability to do that, and not in large-scale studies. So this is emerging technology that is moving forward, and it will enable us to see, and we can see now, what are the most important network? When do they come on the developmental line? There are now complementary measures to study brain anatomy and, you know, function way, like years ahead of what I thought when I started, and we were just really measuring it, sitting and measuring it and circling it. The way it's done now on thousands and thousands of scans with data deposited in repositories, and people able to pull in like the Enigma for various disorders on thousands. So science has become more rigorous with higher reproducibility. So the time that we had 20, 30 scans of people with, for example, schizophrenia in control, and we said, whoa, those days are over. Ned will never publish a paper like that in the American Journal of Psychiatry. So it's the great opportunities, and that's the progress that we have made. Another challenge is that maturation is protracted. I noted that people with schizophrenia don't present till the late teens. Very hard to get to retrospective information. So the prospective longitudinal studies are critical, and we have to follow the kids, because when they're young, it's so dynamic. we don't know. The truth is that we don't know. And the fact that many of them have multiple diagnoses, anxiety, NOS, ADHD, it's not because mental health professionals don't know, it's because that's how the brain works. And then we have to follow and see how it unfolds and what relates to what. Behavior is complex. I mean, think about all the senses, the things that we can do and multitask. It's amazing. What do we focus on? So here you might focus on emotion, but emotion, I mean, it relates to regulation, it relates to perception. I mean, it's all together, although when we dissect, rightfully so, we might focus on one area and then see how it relates to other. And it's challenging to put brain and behavior together because you need to obtain data on brain and behavior and have norms of typically developing that are age, sex, demographically related, so that we compare not apples to oranges, but apples to apples, and we take into account and we are responsible in the conclusions that we draw. And neuropsychiatric disorders that are the expression of G by E, deviating from normalcy, are multifaceted. So that comorbid, as I said, people have multiple diagnoses. They impact other medical disorders. It's one body. So the separation between, I mean, it can work, but not in reality. I mean, the way we feel impacts how we take care of ourselves, and it's associated. Depression, for example, with diabetes. It's not coincidence. So we need many times to treat more than one disorder. There's high morbidity associated with it, high toll on individuals, family, mortality, and stigma. So I still have to, when I work with families, when there's an individual with schizophrenia and his sister, for example, is getting married, the parents don't want to take him, the affected person, to the wedding. So he will not be at the pictures, the family pictures. It's as if he's dead. And I say, no, we train him to go to the wedding. He's part of your family. Accept it. What steps are we going to take that the wedding will be successful and he will feel I'm part of the family? Think about it, that some families ship them away because of stigma. Okay, so what approach have we taken? And when I say we, it's a village, and others are taking, but I can share with you what we have done, but it reflects on progress in the field in general. Comprehensive data gathering. So when the geneticists are looking at collecting data on thousands and thousands of case control, it's a dichotomy. They meet criteria, DSM-5 now, or they don't. There's much more to people beyond the DSM-5. The DSM-5, 6 will be soon on its way, is critically important in medicine. There's no question about it. But when we want to understand brain and behavior, we have to go beyond it. So we have to gather data comprehensively. There's to be multidisciplinary. No matter how good a person is, you can do it yourself. It has to be a group of people with converging expertise, longitudinal, the child developmental trajectory, working with the families and globally, checking yourself against finding. Is what we learn here similar to what collaborators in not just Europe, but South America and Africa find? What is the common signature of a phenotype? It's important to know. So if we discover something in the US or countries that have more resources, we can generalize it and try to help as many people as possible. So we translate to care. A few examples of how we've got the move from phenome to genome. Some of the slides are busy. I will highlight what I'd like the take-home lesson to be. So this is an example of schizophrenia, the disorders I've studied throughout my career. So we know that in the general population here in the bottom in green, it's about 1%. When it's dual mating or monozygotic twins, it's 50%. Note, at best circumstances, 50% genetic. It's high, it's highly heritable. And in the earlier studies, and now the samples have doubled in recent publication, common variants. So we have learned that there are some alleles that you can see, it's a Manhattan plot, the green on top above the line are significantly associated with schizophrenia and are not seen in controls or similar demographically. And those genetic findings are related to what we know on neuronal development, on brain maturation. So it makes sense. Notably, they're not unique to schizophrenia. There's overlap between schizophrenia and BP, bipolar disorder, especially the one associated with psychosis features, suggesting that psychosis cuts across disorders, and intellectual disability. So people's schizophrenia was termed years ago, previous century, dementia precox, characterized by cognitive deficit. So some of the findings in genetics are similar, also similar to some in autism spectrum disorder. So emotion deficit that are seen in autism spectrum disorder are seen also in people with schizophrenia. So there's some overlap, and we can learn by comparing disorders or pulling it together. I noted before that the genetic findings in psychiatry, psychiatric population, have been a dichotomy, not affected or affected. The real world doesn't work like that. There's always people, it's a normal distribution, people with low liability, and most of the people are with medium of liability, and when passing a threshold, a specific threshold, they will manifest the full disorder. So the approaches currently are to look at a behavioral domain or a brain domain, brain behavior, dimensionally. So to take people who don't have it, have a touch, or manifest full disorder. Most of NIMH, National Institute of Mental Health funding today, in the study of schizophrenia, with big network established, is for people with sub-threshold psychotic symptoms. Before the blue, before the break, and trying to see, can you identify reliable biomarkers that will help predict who is going to transition? And that's true in other fields of psychiatry as well, across diagnosis. So now I'm going to turn to what Ned has mentioned, the Philadelphia Neurodevelopmental Cohort. So it was supported by ARA funding. Previous President Obama tried to stimulate the economy and put in across in NIH Institute requests for funding. And one of them was on neurodevelopment and genetics. Looking at it, we said we had an opportunity. Children's Hospital is on campus. It's part of the University of Pennsylvania. And we collected over a little bit more than 2 years, 9,400 and close to 80, close to 10,000 kids, age 8 to 21. All in person, nothing virtual. With the pandemic, a lot has been moved to virtual. We got to know them and the families, and for follow-up turned out to be important. So over 50 clinical research coordinators, bachelor's and master's level, were trained quickly to administer an adaptation, a computerized adaptation of the case studies. That is a structured, semi-structured interview for kids, okay? But you can do it for young adults with some adaptation. And we deployed a computerized neurocognitive battery in the middle that was Ruben Gur worked on it over the years, based on functional imaging studies, tasks that show specific activation of a circuitry that I will illustrate in a minute. Put it on a laptop, and you can go to a kid's home and do it, okay? And if they came to the lab, it's done in the lab. And 1,600 multimodal imaging. We'd have done more if we'd have gotten a bigger budget, but it is what it is. All of them were previously genotyped with permission to recontact. So one thing that I've learned then, and that all of you should know, if you don't know yet, in an informed consent, always get permission to recontact. If you don't have permission, it's very hard, okay? So you get assent from the kid, consent. Most cases was mom, sometimes mom and dad came in or invited us to the home. And when we recontacted, we told them, we'd like to follow you. We don't know when, it depends on budget. It's not because we don't care. We need a budget, we need a grant to support it, okay? So always ask for permission, and I'll show you how it paid off. And you ask for permission to have access to the electronic health record. The field has moved. There are no more. You go to a doctor's appointment, there are no more handwritten notes. It's all electronic. And you can have big parties with AI, with machine learning. Amazing what you can do with it. But you need permission. So permission for both. And that's what we had for all of these kids. Okay, so now moving back to brain and behavior. What did we learn? We learned that there is a P factor, and I know it's debated in the literature, but sometimes you have to come up with a number of how overall, what is the burden of psychopathology? So in a bifactor model, we came up with overall psychopathology. So there's one number that you can say, this group of kids have a big burden. This group of kids don't have a big burden. And then you can begin to dissect it. And what does it fall into? The expected categories. Fear or phobias, kids are characterized by that. Anxious misery, anxiety, depression, that many times go together. Psychosis, and externalizing behavior. So now we're parsing this, different investigators pooled, and these were all junior investigators at the time, followed one of, depending on the interest, concentrated on one of the groups, for example. These are the stimuli that we used in the PEM, computerized neurocognitive battery. It measures accuracy and speed, multiple domains from executive function, complex cognition, learning, and for the interest here, emotion. Oh, sorry. So it's emotion, recognizing emotions, differentiating between intensity of emotion and age differentiation as a control. These tests were derived from studying multiple people in collaboration with the University of Pittsburgh, multiple people, same scanner, same protocol, realigned with these tasks. So we knew the circuit to be validated. That's how we proceeded. And now I will quickly go over to show how studying individuals with subthreshold psychotic symptoms, you discover that the brain behavior signature exists before psychoactive medication touched their lips. They were all naive to medication. And before they met criteria for psychosis. The train left the station before we ever meet individuals who present with psychosis and likely for other disorders. The step by step by step. And when we come to see them clinically, the developmental trajectory is bent definitely for psychosis. So what we see here, these are all people who were fellows at that time, now the faculty with us. Look at the working memory. One of the most well-established findings relating working memory to dorsolateral prefrontal function, showing multiple deficits, multiple studies with meta-analysis, impairment in schizophrenia, poor performance, less activation. On the left, PD, typically developing, several hundreds of them. On the right, PSs, psychosis spectrum, subthreshold psychotic symptoms. And what you see, they have poor performance, I don't show, but look at dorsolateral prefrontal, they have less activation. Less activation is associated with poor performance. And that's on the right is PS minus TD. So you can see less activation in the PSs. Look for emotion identification. It's the same group of people. They've gone in the functional MRI, both tests. And you can see that the visual cortex, they're looking at faces, highly active in both groups, TD and PS. But look at the amygdala in the middle, much more active in PSs for threatening faces that are perceived as threatening, angry and fearful, okay? So there's greater angry faces are perceived as more threatening. So you can see greater amygdala activation. We and others have shown it in people with schizophrenia. So I'm showing it to illustrate that the brain changes, the signature associated with the disorder is evident before. And also if we look at developmental trajectories of the cognitive, chronological age to neurocognitive age, predicted by performance, blue line typically developing, green line, other psychiatric disorders, primarily anxiety, depression. Red line, psychosis spectrum, the touch of psychosis. And you can see that at a young age, we started age 8, the psychosis spectrum were impaired already, these kids. This is a cross-sectional study, but in adolescence, they become differentiated from the PSs, other psychopathology. And in the 20s, when we finished the study, they were up to age 21, they go south. And I will show you what happens to them when we follow them. Okay, so this is, I've illustrated so far how you can study large-scale multimodal data and that it gives you useful information for a specific disorder. Because we are interested in psychosis, we focus on psychosis. You can do, and some people are now doing depression, anxiety. We're now doing externalizing behavior. We are working with the city, with the district attorney's office, for example, to find out what happens to the individual, the kids then, there were more males than females, with externalizing behavior, ADHD, opposition defiant. How many of them ended up over 10 years later involved with the law? So we are working with the lawyers of how to do it anonymized. So we don't, that it's done an honest broker approach. Okay, so the people who analyze the data don't know anything, and it's worth the effort of doing it, but we can see how early behavior predicts. Environment. 2010, one of the good things the US is doing for research is, in addition to funding, is census. So at 2010, when this study was on its ongoing, there was a census. And in the census, we get block, by zip code. We have zip codes for the people, for the kids. Block by block, what % marriage, racial distribution, income, below 40, all of what you can see in this table, population density, etc. In blue is what we collected during the KiddySats applying. So we looked at, have you seen anybody killed or hurt? Have you been attacked? Have you been part of a natural disaster? Describe. Sexual assault, bad accident, threatened with a weapon, okay? So we can come up with a risk factor of an environmental risk factor. We want to be at the table with the geneticist. So we can give numbers that will lead to G by E. Rather than say good environment, bad environment. If you want to replicate, you have to come up with a standard way of doing it so other people can do it too. All right, now it's going to get very busy. I will highlight everything that I've said so far that motivated what we were doing and other investigators in the field. What does it show in this sample? All right. Here are symptoms, and the symptoms are mood anxiety or anxious misery, fear, phobias, externalizing behavior, ADHD, opposition defiant, and psychosis, okay? This is SES. These are all its standard symptom score. In the black line, the 0 line is normal SES. It's not a low SES, okay? In the orange line is low SES. And if you ask, what is the contribution of SES to psychopathology? Manifested in these clusters of symptoms, in each of these clusters, it's elevated. A little bit. You can grow up poor and be in a supportive environment and be perfectly normal as far as psychopathology. So it's a little adding a little bit. Now look at traumatic stressful event. 0 is the fortunate, no trauma reported. Blue, 1, major traumatic. So the traumatic event, I emphasized it, it's not like mom was not nice to me. Well, a real trauma, where you're afraid, you watch somebody die, you're attacked, or you're fearful for your life. It's real trauma. Red are 2 traumas and more. And now look at the distribution to psychopathology. Major across domain. So for those of you who are going to be engaged in mental health care, always ask about trauma. I was telling that in psychosis, I didn't initially. Why? Because it was so overwhelming to see them acutely psychotic. That's what I was doing, working inpatient, acutely psychotic, or transfer from inpatient to outpatient. Well, I mean, it was, I couldn't sit and get a comprehensive history from them. Just have to make sure that they're safe to themselves and others. But now we ask each one of them, and there's trauma galore. If you don't ask, you don't know. So we have to ask about trauma. And you can see across the board, including psychosis. So some of the people need to be treated for trauma. And there are methods, behavioral intervention for trauma, that work for psychotic people as well. We didn't do it before these findings. Okay, so you can see it for female and you see it for males. Okay, so the moral, the conclusion here is a traumatic event impacts psychopathology more than socioeconomic status. Sometimes it can be a socioeconomic status and trauma. That's a double whammy. So the risk is higher. Okay. Now, look at puberty. Recently, there's a line of research in mental health that has been looking at the impact of adversity on puberty. You sort of have to grow up fast to become sort of an adult, take responsibility when you have to. It's not like you're not allowed to be a child. Turns out, I just heard last week, there was a meeting of cancer in San Diego. And one of the plenary presentation, I have to find it out see if it was published, but one of them was a hearing on NPR, was on how stress, let me go back. We see more that some cancers that were associated with people above in the 60s, 70s, are now seen in people in the 40s and 50s. And the question is why? Stress. So in cancer research, they're talking about the impact of stress on aging, that our body ages, okay? So it's not a psychopathology. We know the heart, stress impacts the heart. So any help that we can provide based on good research is going to help not just the psychopathology, the psychiatric, it's going to help across disorders. And here in our sample, what you see, it's busy. So I'm just saying SES and puberty, we measured puberty. And you can see that the red, this is benign, is the orange, the red is low SES, accelerated puberty at a younger age. TSE, traumatic stressful event, is the same. So lower socioeconomic status and trauma are associated with accelerated puberty, aging, premature, and that's important. And now if we look at the cognitive, what is the impact of the cognitive performance, the battery? And you have the domains, executive. Can you hear me?
I think you can turn on the.
Now you hear me better, right? Thank you, thank you. So here are the domains of executive, of cognitive domains: executive, memory, complex cognition, social cognition, the emotion type tasks. And what you see is that people with low socioeconomic status perform less well. They're less exposed. There are no books at home. There's no early education is important. So low socioeconomic impacts cognition more than psychopathology. Now, if you look at traumatic stressful event, TSCs, you can see that they are not worse off. Some specific for complex cognition, I'd like to see it replicated, but they're not. So traumatic events impact psychopathology, low socioeconomic status impact cognition. All right, it's busy. I want to just point out one thing when we move to imaging. It's related to parameters of brain. Remember I mentioned that there are different parameters to brain. So here is, for example, if you look at brain volume, is low socioeconomic status, less stimulation, less opportunity to learn. You can see that brain parameters are somewhat decreased, somewhat. If you look at TSE's comparison, it's for specific brain regions. It's not across the board. Similar for gray matter density. Gray matter density is decreased for socioeconomic, but it's increased with more trauma. It's not a finding that is easy to explain, but we are trying to replicate it in other samples that have undergoing imaging studies now. Okay, and now if you look at adversity and the brain aging, just focusing on that, these are the age groups, and you can see socioeconomic status affecting males and females, and adversity, traumatic stressful event, earlier aging. Now we have followed the people. I'll illustrate the data from one protocol on psychosis, risk and resilience in psychosis. We've taken the kids who have participated in the study, and some of them were TD, no psychotic symptoms at all, and some of them were PS, a touch of psychotic symptoms. And now we follow them 10 years later. So we have been in touch over the years, but now they're coming back for a full protocol, okay? And most of the people with TD, the green, TD, remain TD 10 years later. Some of them become other psychopathology, anxiety, depression, usually. And some of them become PS, psychosis. Similarly, for OP, other psychopathology, some remain, some are TD. Yeah, they had the symptoms they remember, or they might not even remember that they said so. They say, oh, it was a tough time. Grandma passed, things like that, that we get from type of comments. Some of them become PS. And the PS, majority PS, and some have transitioned to full meeting criteria, DSM criteria for schizophrenia. And some of them are TD. They're fine. They're resilient. So now we are looking at baseline and over the years, what protected some of them and what tweaked the developmental trajectory. And one of the things that I can say is that its cognition is very important. Those who recovered, there are no PSs anymore, had better cognitive functioning to begin with. So advancing cognitive skills is important, and those with lower opportunities will be more vulnerable. Okay, it gets busy, but I will just show, this is a scatter plot of the same, of what I've just said, looking at the change in cognition in relationship to the change in global functioning. Both was measured at the same time. And these, the greens are the typically developing. With age, with age, you expect improvement. And you see that those who improved and remained TD, cognitive improvement with age, and functional outcome. Those who became other psychopathology, less cognitive improvement with age, and the outcome is more stilted. Those who became PS are the lower line. Same applies to the other psychopathology. Those who, are fine, show more cognitive improvement and better outcome. Same for the PSs. So cognitive changes are associated with changes in psychopathology. Okay, so to summarize what I've said so far in common variant is that there are tools now, especially in longitudinal studies, to identify major domains of psychopathology, major domains of cognition and emotion, and follow individuals. The follow-up now can be done virtually. So we have had a study that we showed that doing it in person or by trained people, coordinators, or virtually during the pandemic is the same. You get similar data. So it opens up the opportunity to do it. And now what we are doing in psychosis, talking to Dr. Kalin about how to bring research and clinical, which is very difficult to do. In our psychosis clinic, when people call in for an appointment, you assess why are they calling, etc. We refer them, the whole thing, insurance, the whole thing. They seem to be okay, they can wait for an appointment. We tell them, while you wait for an appointment, we want to be prepared when you come in. And we connect them to getting a screen of psychopathology and the cognitive battery, okay? Virtually. So when they come in, the data is available and we can see how many problems do they have and what is cognitively, what can we do about it? Because for young people, we advise them about, should you drop out of, take a leave of absence from college? Many are college students. They're applying for jobs. What will be the best job for you? What should you do? So we try to take them off rehabilitation, advising them for how do you move beyond the psychosis when you're stable and doing well with therapy, family support, medication, etc. So it's clinically translatable is what I want to emphasize. We do research not for publications. I mean, publications are nice, but after a point, you do it because you want to impart the knowledge. This is public money. You want to impart the knowledge to benefit as many people as possible. Okay, rare CMVs. Rare CMVs are mutations that take place. You're born with them, so they are de novo mutation. Sometimes you can inherit them. Sometimes it just happens. And just to show that, if you look here, this is the common variants, alleles, that have been found in schizophrenia, case control. So for example, C4 is related to inflammation. A lot of the studies now are inflammation and schizophrenia and depression and other disorders. It's not unique, it's not specific. But if you look now, look at the green points of rare CMVs, they're rare. But the odds ratio is much higher. So knowing that, it appeared in the literature, Children's Hospital on campus is the largest sample of and specialty in 22q 11.2 deletion syndrome, 25 to 30% increased risk for schizophrenia. So you can see the kids and evaluate them prospectively because they come back and assess when do the psychotic features emerge, what precedes them, and how can you treat them potentially before, and what is similar. There's more similarity across everything globally between people. I mean, if you get and you study people, a lot is the same. There are, of course, cultural differences, but a lot is quite the same. Also for rare CMVs. I'm more impressed with similarities. And how can you learn from them to benefit the population at large? So, busy slide. by Aaron Alexander Block, who has joined us 3-4 years ago. And he looked at the entire PNC sample for rare CMVs, okay? And what I want to show you, these are here on top are the cognitive measures, all the cognitive. Look at those who are significant, the top. You can see that in psychosis, those who are impaired cognitively are more likely to be psychotic, okay? And psychosis is more common and also externalizing among those with rare CMV. So it's an opportunity to probe, to look at the sample and follow the rare CMV. And this is the mutation that takes place in one chromosome. The Q part is deleted, about 40 genes are deleted. So you can, the geneticist studies, it's like a domino effect among the genes too. But what's important is the psychiatric presentation. They have developmental delay, ADHD, anxiety, and psychosis, late adolescent, early adulthood, just like the general population. It's a window. There are mouse models of 22q deletion, and you can, from the participants, you can establish cell lines when they come in, they get blood diagnostically and for laboratory, and you can establish induced pluripotent stem cells. And then in a dish, you can try and manipulate and look at those. What we look at is those that years later develop schizophrenia and those who do not. And one of the findings that we have in others, replicating in others, that it's the mitochondria, bioenergetics are implicated, that people who do not develop schizophrenia compensate through the other allele, and people who develop schizophrenia do not. So it's an opportunity to translate it potentially to therapeutics, novel therapeutics. Look at the distribution of symptoms. Color-coded, you can see that in kids, in the deletion, more anxiety, more ADHD, and some psychosis features, but they were young at that time. So now when we follow them over the last decade, more psychosis is emerging. Remember, late adolescent, early adulthood. And the factors of psychopathology are exactly what we see in the general population. That's why I say that it clusters similarly. So you can see there's a psychosis, ADHD, mood, and anxiety in this population, and the overall psychopathology, the P factor. And what they have is similar in schizophrenia. Negative symptoms, positive symptoms, and then some of them, more in later adolescent, early adulthood, pass the line and meet diagnostic criteria. Cognitive, we compared them, blue line typically developing. These are all the cognitive domains. Blue line typically developing. We compared them to developmentally delayed without 22q deletion. It can be developmentally delayed without a mutation. To see how much the developmental delay. And you can see that in most domains, they are close to developmental delay, to the development delay, except two. Complex cognition and social cognition. They cannot read emotions on the face. 20-25% of them have autism spectrum disorder. All participants have IQ above 70, they can perform, they understand, and they are engaged. It's important clinically because many times parents, we as humans, we communicate in our face. With, you look at somebody that say that really mad at me, and you try to figure out what did I do wrong as a kid, what's happening. Now I can tell the parents, they can, when you come into the room and you're upset with them and you show it on your face, you son or daughter, they don't read your face. You have to tell them. They don't read faces. And the parents look at their performance on the face. They say, wow. You have to tell them in a nice way, I am, and you have to repeat it and make it simple. So the study of emotions, and you see it in those with autism spectrum features, but those without. So it's a major finding in this group of people, okay? It went global when it's rare, and I mentioned how important it is to collaborate. within this country and other country. It went global, and guess what? The same results you see across the countries. And it's South America, Europe, Canada, and North America. You see the same results. So blue, psychosis is later on in life. Exactly what we found. And what we found in this large study is that cognition, here's simple number, IQ, is declining cognition, the blue line. This is the full scale IQ. People followed, studied twice. And you can see that the people with psychosis have more decline in full scale, and its top line, it's in verbal abilities. So declining cognition. Many times when the parents say they're not doing as well in school, precedes, and that's what we see in psychosis too. Cognitive decline suggests that the brain processes are pushing away from the normative developmental trajectory. It's a sign. And they have multiple MRI deficits on multiple domains. There's now an Enigma network that all the people have been collecting data in separate R01s are contributing. So you have large samples, and you can see the multiple parameters of brain anatomy. If it's volume, cortical thickness, surface area, gyrification are impaired in 22q deletion, and those with psychotic features or schizophrenia already more impaired than those without. Same thing for brain connectivity. Similar findings to what you see in individual with schizophrenia. Those with 22q deletion and depression, anxiety don't show the same. So it's more unique to psychosis. All right. And now with the library, or with sharing data that is available, we can look at the cerebellum. So we can look at structure of the cerebellum that are also the cerebellum. It does not coordinate, not as coordination. There's a role in cognition as well. So you see it across all brain parameters and region. Now we follow them. I have a few more minutes, right? Yeah. So now we follow them. It's very tough to do, but I think that it's essential. You see them cross-sectionally, and it's very interesting to see them years later. Now, you know, it's a young man and a young woman sitting, and you saw them a few years ago, and they were kids. You know, it's very different, and it's hard to predict. So because of our interest in psychosis, and we are now expanding it to other psychopathologies, is that we look at all domains that are examined in the study of psychosis in the regular population, in common variant population. So you look at positive symptoms here, you look at negative symptoms, you look at disorganization, general symptoms. General symptom is like sleep. We measure sleep as well, and global functioning. The red are the individuals that were young, they were kids at that time, who entered as PSs. They had the psychosis touch on SIPS. It's a structured scale interview for psychotic symptoms. So we adhere to what everybody is doing in the field so that we make sure that we can learn from each other. PSs are pinkish, reddish, and no PS. They have deletion, no PS. Look across the board for domains that are measured in psychosis. Those who started at PS, you can see over the years, elevated the positive symptoms. The negative symptoms are there. General symptoms are there. And functioning is below. So positive symptoms at a young age is a sign of persistence of the symptoms and poor functioning, okay? Now, if you look at neurocognition, remember both were measured in all the kids, is what we can see is some domains, look at memory right here, doesn't change much. Doesn't change much. And doesn't differentiate the positive and the negative. But executive function and social cognition does. And I will show you more here. So here is when you look at positive symptoms, negative symptoms, disorganized, general, looking at the slopes, and GAF, Global Assessment of Functioning, in the clinical domain, you can see that the PSs, for most parts, are more impaired than the no PS. They have no psychotic symptoms, and that the GAF is lower. If you look at cognition, both groups are impaired. The only difference is in, if you look at across domains, and now you look here, it's in the executive function. And if you break it down, it's in working memory only. So working memory, so if we have to make it, if you want to cut how long it takes us, we zoom in on working memory. Remember that when I was showing the slide on the MBEC, it's a working memory task that is associated in schizophrenia with impaired dorsolateral prefrontal activation. We see it in the kids with subthreshold psychotic symptoms and the deletion. So it seems that it is a marker of poor activation that is associated with more psychotic symptoms across the two populations, common and rare variant. It's complex, it's trying to see what drives what. And it's cognition, structural equation modeling, that cognition drives the clinical change significantly. So it's cognition first. The changes in the brain, that's when I said the train has left the station, and it can be a few years between. I mean, it's a gradual process. It's not overnight. It's a stumble over a stumble over a stumble until it manifests. All right. Example, I'll give you an example of how electronic health records are helpful. The kids, before we ever met them, go through a standardized language evaluation by a clinical linguist who assesses children at Children's Hospital with developmental disability. Everybody gets a standard exam. Never met this person before. She's doing it all the same. She's very nice, wants to collaborate and be helpful. So we went to the electronic health records. Now, years later, we assessed them and said, they're PS or not PS. We did other things too, but for this purpose, a focus on psychosis. And what you see, the PSs are green. Each participant is a dot. And you can see that if you look now at the line, the green lines, is those with PS assessed on language years before, performed more poorly years before compared to the non-PS. There's a big literature on language deficit retrospective studies of kids with psychosis. There's now natural language that is being done as part of ProNet, recording, looking at the literature and look at the health record and looking forward. We have the methods now to pick up in large populations because the test measures are there. It's all in records. So it's an opportunity. You can do multiple PhDs just on looking at connecting with people who have the techniques and with good electronic health records. It's very helpful. And it's clinically translatable because we can pick them up before and maybe begin early intervention with them. Okay, so to move forward, what do we need? We need convergent approaches. We need large-scale collaborative studies. Small samples will not do it. And when we talk about phenomics, there are multiple measures of phenomics. And we have to consider in each study what is the major hypothesis. But if it's not a big burden, it's helpful to collect a little bit more so the field moves with us. Once you invest, it's important to benefit. So in psychosis, what do? Clinical high risk. First episode, people are presenting psychotic, but looking at their lifespan. And also to integrate the clinical care. I gave an example. We now apply our research trimmed battery that underwent adaptive testing, and NIMH supported it. So we can get in 45 minutes a screen. it will go into the clinical records with their permission, so that we will be prepared for them and try to dissect the heterogeneity. And they will undergo MRI clinically, and we can see a special sequencing for psychosis, step by step. And for gene-first approach, it's complementary approach, because if we can gain better understanding on mechanism, it is likely to be generalized to the population at large. And if there are novel therapeutics, others can benefit as well. And the last is the intergenerational. So for clinical phenomics and genomics, we have the tools in between and measures of environment, neurobehavioral parameters, can be linked to non-human studies, multimodal imaging, as an example, iPSC, or it can be organoids, okay? And I didn't mention EEG that is being done, MEG that is being done, multiple tools. So I'll give you 3 examples. One is the intergenerational longitudinal effort. The other is large-scale sets in electronic health records of clinical MRI can be used. And facial dysmorphology. Face developed first, and I will illustrate. So, intergenerational, it's the G and E when we are born and we grow up, as I said when we started. So, assessing adverse experiences, appreciating the impact of systemic racism and being perceived as different by others, leading to discrimination, and mental health disorders in the family that are a burden to the parents and to the, even if it's a sibling or a parents of the parent, when it's a burden that we need to consider and try to help. So during the COVID, we formed a collaboration with obstetrics before the COVID. But the COVID was a sort of a natural disaster that was stressful for all of us. And we followed pregnant women who came, presented to obstetrics, and wanted to participate in research and follow about 1,000 women through the delivery. And we have documented discrimination. About 40% were non-white discrimination against them, worry and anxiety about what will happen to them and their loved one, and will I be able to support my child? Will there be money for food? I mean, really existential type of worries. And then we followed them, and now the kids are coming for the 4-year follow-up. Some of them were born during the pandemic. And you can see, we can assess longitudinally and developed methods for assessing the kids, and developmental psychologists are great at it, virtually. And we buy everything through grant support and send it virtually and record it and establish reliability. They don't even have to come in at a young age and during the pandemic. So with this, we have mom and dads and child, all right. The Philadelphia Neurodevelopmental Cohort is reproducing. And I was mentioning that one of the people called in for the emergence of psychosis study, told us she cannot do it. Yes, she remembered. She's pregnant. A light went in my head. I said, wow, she's pregnant. What about the others? We began to contact. And we have not contacted everybody, but we have already 1,000 kids. Offspring of moms, now we are contacting dads too, who are cared for at CHOP. So we have in the health record everything you want to know about them. And they delivered at Penn Medicine. So we have everything about the obstetrical record, so we can connect the two and give the family ID. So they are beginning to come in, or virtually, depending on the age. And they are aged from infant, some of them will be born soon, to age 4, 5. So we are starting with 3 to 5-year-olds, where there are more tests and we are more ready for them. So intergenerational is critical. The other one is Aaron Alexander Block is doing it. It's a new initiative at CHOP, looking for clinical MRIs. There are thousands of clinical MRIs, thousands. We always look at health records and we say, well, it's health records. What about MRIs? They're in the health records. Sleep studies, they're in the health records. So we tackle the entire health record in addition to labs. Okay. And this is a demonstration project, NIMH funded, that see, can we process the clinical MRI de-identified? There's a honest broker, we make sure, no name, all removed by radiology. We pick up those that had no findings. The neuroradiologists read them, they were referred, no findings. And then they're processed and segmented. And it's a 3T clinical that is the same 3T for research. It's just a clinical sequence, but it's good. So you can see it here. And now you can see here, validating it, clinical controls and research controls. He did first a study, collaborative study on 100,000. He was a PhD student at Cambridge. So he did that there, published it last year. And you can see that the pattern, this is the age, the pattern is very similar in clinical controls and in research controls. And here you see the distribution by age. And this is the reason for referral. So developmental disorder is suspected. They have clinical eye or vision findings. They get a brain MRI. Headache, other, seizure suspected. And you can look here on brain parameters, the centiles. There's no evidence that there's a bias in the measures based on the reason for clinical referral. So the first order was like similar to, in the posters yesterday, I first needed to validate my measures. Exactly. So validated the measures. And now, out of these kids, you know, in last we meet, how many kids I asked at 22q11.2 deletion? 168 kids at the deletion. Before we ever met them, we were doing them with research, subsample, older. Language, how does the brain parameter at a younger age relate to what we know about them now? So it's an opportunity to incorporate, to get 168 kids with the deletion for MRI. It's a 5-year grant, very hard to select. They don't move. Oh, you can sedate them in research. They have to be older. It's clinical. They're sedated, if need be. So we need to think about how can we pull together to learn, because this is independent of what we assess now. All right. And the last is to go to early development. That's a work of David Rolf. I'll show his picture in a minute. For those of you who have ever seen an ultrasound of pregnancy, and you look at it, and the face comes on both face first. So, right, I just yesterday looked at my son, his wife is pregnant, and this, the face I see there, and there's the nose, the eyes, the lips, the face first, early in development. So, we can see this when this comes on board, it looks like a little bit like a frogish, and this, the maxillary, and now at 6 months, the mandibular, it's a face, okay? So, and we know what layers do they come from? So we know when it occurs in development, right? We can tell, we know, because it's like a programmed plan of the development of the fetus with head first. So we know when and what neurons, we can tell it from studies that have been done before, so we can connect with the basic. And these are 2 programs that I will illustrate, and this is David, the paper he just submitted, and he is the subject, so he doesn't have to ask permission, has permission from himself, that's how we solved it. And there are ways of automated measurement of the face, and you can delineate what are the regions that you want to measure that are linked to the emergence of the face developmentally. And then you are able, and he's doing it in 3 populations, 22q deletion. We assess them. He doesn't know them. He's a behavioral neuroscientist. So he does everything on his own. That's a principle also. The labs are doing it separately. Then we merge it. So he doesn't know what we do. I mean, he knows what we do, but he doesn't know the values. And we know what he does, but we have not the values. When the study is over, we put it together, combine our data sets. So he's doing 3 groups, 22q11.2 deletion, typically developing, and people at clinical high risk for psychosis. And what you can see is that you can identify the area under the curve for people with the deletion. It's less strong for people who do have PS without a deletion. But in other studies, I don't have time to show, that are in this paper, specific regions for the clinical high risk that relate here, philanthropy, a mobile closer to the eyes, are different, and you can tease them apart from those who are typically developing. These are quantitative measures of trying to tie it in to development and pull together. So overall, if we want to transverse the line of research from basic, translational, clinical, and generalizing to the population, we need to study the genetics, the environment, that shape our brain, and the product of the brain is behavior that can be linked to brain, and symptoms that we try to link to brain across domain, and they influence functional outcome. So it's a long road, but there are greater and greater tools, ease of communication and obtaining measures, so you have to go get them. This effort has been done by many, many people. These are the current, supported, who worked really well together. Other collaborators from Penn and outside Penn over the years has been supported by NIMH, a grateful family with 2 family members, 2 siblings affected by schizophrenia, and the Lifelong Brain Institute, Libby. Thank you. You want to take questions?
Thank you so much. Breathtaking, really a great start to the morning. And I'd like to invite Stephanie Ward and Ryan Hampton, who will lead us in the discussion.
You want me to stay here?
Yeah, you can sit down.
Ah, thank you.
Just turn on your mic.
It's an honor. Thank you so much.
Oh.
Oh, it's green.
Ah.
Testing?
Now it's on.
Perfect. It's an honor. Thank you so much.
Thank
You've outlined several challenges and potential steps forward for conducting multifaceted studies of cognition and social and emotional development. And as a trainee myself, I'm wondering what you think the most valuable elements are to emphasize in training clinical scientists who want to conduct either basic or translational work that can move the needle moving forward?
It's a great question. You always have to start. I'm reflecting of how I started and what I advise junior people, trainees, graduate students, fellows, and junior faculty. You know, this is a culmination of a lifetime. So it's like a story that is based on what we did, what I'm familiar with, and what motivated it. But you always start at a step. It's not that you can start with I mean, it's cumulative effort that involves many, many people. So when, as a graduate student, I mean, you want to be done in a reasonable period of time, 4 years, sometimes, you know, about, and you choose a laboratory that is based on your interest when you come to campus. So that's one, and that you see where they're at. I mean, they were making progress and studying probably even before you were a graduate student. As time goes by, maybe before you were even born. So where they at? What are the a balance between the lowest hanging fruit that you will be able to learn, understand, master, and publish so that you move forward, and then thinking of how do you align with the next steps so that if there's the next stage of the study, an experiment, talking to the mentor is, this will be really nice to do, and there's a gap in the literature on that, and we can do it in a reasonable period of time, that will be the next step. So I think a dose of reality, I mean, I like to dream, but a dose of reality is important, and to work closely with the mentor and sitting, what is the next step? What will you move you in the lab? Because sort of, you pass through the training period, you pass through life. And what do you want to accomplish? And graduate school is a short time. So I will say start simple, but be aware of where the field is. So by going to meetings, learning to present, reading papers, and being part of a group, that because it's too much, and listening, then you can collaborate with somebody. For example, there's one of the people in our group who, a child and adolescent psychiatrist, who focuses on the environment. The imagers, now they say, well, you know what? The environmental risk factors, bring them on. We'll put it together with imaging. They collaborate. Does he understand what they do in imaging? Yes, he does. He's intelligent, he reads, he can understand, he can present. Can he do it himself? Absolutely not. No, I will not get him close to processing. Can they do the environment? No. I mean, they're good with computers, but they don't, it's not. He has the sense for, by seeing patients and reading the literature, what is important. So it's team science. That's when I said that. So who is on your team? And many times the mentor will be able to say, he, she, they have a different perspective. So to be open-minded about it. That's what science is really about. Did I answer it?
It did. Thank you.
Yeah. Is this working? Check. Can you hear me? Okay. Like I said, this is a really incredible lifetime of work. Like you said, it really tells such a nice story. Maybe not a nice story, but a very clean, very parsimonious, really well-researched story. And so one of the big takeaways from this for me was that this work clearly has a ton of predictive power. Taking all these longitudinal studies and seeing how you can predict things down the line with baseline levels and all these kinds of different trajectories. I was interested in how now do we apply that knowledge. Like you mentioned briefly about if we can know that there is this predisposition early on, there's a chance that we could do something about it. What kind of approaches would we take in addressing that proactively?
Okay. I'll give it a try, but I will say that unfortunately, the funding that is provided by the people who fund, if it's the government or local agencies, is not commensurate with the needs. So there are significant portions of the populations who are left out. And health equity in the United States that I'm most familiar with is a major obstacle. So populations are being sort of, they're not what I call not at the table. So I think I see part of our role as health professionals to put it at the table, so that not to take away from cancer, diabetes, whatever. Everything is important, but the quality of life and mental health are critical, and everybody deserves it. So approaches are, and some of them are done. Screening early. So for example, there's early screen, at least at CHOP. I've, one of the things is that I've always been, as you were saying, in one place. So I talk to colleagues in other places, but it's different. In children's hospitals, differ too. Every child at age 2 is screened for autism. Every child is screened. It's mandatory. You can go to the health records and look at the scores. So we are looking at it now. And the kids are followed. So you can begin at kids that were deemed at age 2 to look at the distribution, males and females. What else do they have in the health records? And begin to design a study of saying, and the hospital will support it. They'd like to see novel approaches to treatment. We are going to pick up a group of kids. These are the characteristics. And we are going to have a group of kids, including them, those who are coming from more of an adverse environment. So they are at the table. That's in the design of the study. And we are going to contact them because there's permission to recontact. And we're establishing, I'm giving you concrete examples, advisory boards. The people from the community need to be part of it. I don't want to tell them what they need to do. So we have advisory boards for psychosis, for example. What is bothering you most? How my kid will be at the wedding. Did I think about it? No. So listening to the needs of the community and connecting them. And then in treatment interventions. So there are people, clinical developmental psychologists, who are working on language, on self-regulations. So they don't have meltdowns, that advocating for them for special schools, for more individualized intervention. So for the, to do a CHOP, I can go on and on. You can see that that's something. To do a CHOP, a neuropsychological evaluation, they don't support it. The insurance doesn't for many. It's thousands of dollars. For people who can afford it, wonderful. But what about the people who can't? So we generate a report based on what we see. And we say it's research, but this is something that maybe can help you with the school to say that you need a little bit more time. You need more small group. You're overwhelmed in a big group. You become anxious, things like that. So how do we translate it in the clinical setting by showing the data? So showing the data to pediatricians, telling them I'd like one of the people to present in your grand grounds, and hooking with some pediatricians who become advocates for us. So we have, in our training grant, people in pediatrics who are research-oriented, but are wonderful clinicians interested in neglect and abuse. So you look around continuously to find somebody that you can connect with. And I feel our patients deserve it. So to be an advocate, and there's no one way. So by pulling people from other disciplines that are impacted, they see the kids and making it part of the agenda. So, and sometimes when asked, even when there's a lot goes on, I don't want to be on a strategic planning, but I go. Why? Because otherwise it won't be mentioned or will be there. Nobody will talk about mental health. Well, I'm there talking about mental health. I show them this or that, and they say, maybe we should do that. So I wish it would have been easier, but the more people are doing it, consistent effort, and learn from each other, so it can be translated across institutions. And saying they're doing it there, maybe we can, and more collaborations. So if you have something here that is planned for younger people, we'll collaborate. We will train, we will do, and pull the data together. And then you have East Coast, Midwest, West Coast. You make an impact. And also you argue for on the state level. So it's like going to, being somewhat more politically active, but for the mental health cause.
Thank you.
I'm wondering about, you talked about the influence of cumulative traumatic stressful events and how this might interact with other forms of environmental adversity to predispose people to multiple types of negative outcomes, including psychosis, but also other forms of psychopathology. Given the lack of resources to go around that you alluded to, is there anything that you think could be changed within our school systems that might help either screen or support children who we know are at risk for multiple reasons across the social ecology, genetic, environmental, and otherwise?
It's a good question. It's tough. I've talked to conferences of educators, and I asked them, how many of you know that a mother or father of a child in your class has cancer, and it might impact the child because they're diagnosed, they're going through treatment, or the child, might not be able to come to sports activity, etc. Almost all of them raised their hand. How many of you know that a parent or somebody in the household is a psychiatric or mental health? Maybe one hand. People don't want to talk about it. There's a sense of shame, almost like something, stigma. Something is wrong with me, rather than, there are factors beyond me that has influenced who I am, and I'm trying to do my best to move on, one step at a time, be resilient. So it's not there, I will say, unless that's my experience, also with my kids going to school. Some schools respond more to bullying, but even, it's so common, it's frightening to see how kids are being bullied, on social media and in the class. And these are kids who are not with reverse CMV, who are make it part of the curriculum, are not to marginalize, have parents who want to talk about it. It's our kids. So have a community that is engaged and in school meeting talks about it. Advocate for discussing at the child's level, of course, that this makes somebody else feel bad. Things like that, and sort of coalitions of doing it. I know that in some of the schools, the parents are in a network and they talk to each other when this happens outside the school. They monitor and say, this is a topic, my kid came today beaten up. And sort of in the neighborhood where all the kids go to the same school. Has this happened? And should we put in a note together, email to the head of the school? It's advocating, not feeling by being quiet, it will go away. It doesn't. So, and it's hard for the teachers, they'd rather tell the parents that the kid, is too much of a burden and they need to go. Sending them out from the school, they don't fit. Yeah. And especially with a young kid, how do they know? So the ratio of teachers, it's a lot. So, I mean, for people, when they choose a place where they want to raise the family, look at the school system. It's important.
First, thank you very much for your talk. And I have a question regarding SES. So during the longitudinal trajectory, people's SES may move upward or downwards. So do you expect that kind of change will exacerbate or buffer the genetic risks of pathology?
I missed the first sentence part of it. I missed it. Can you repeat the first sentence?
So during the longitudinal trajectory, the SES will change upward or downward. So how that kind of change may
Okay.
buffer or exacerbate?
Yes.
Yeah, and do you find any evidence support that kind of?
So as you point out correctly, that environmental measures change. It's a dynamic process. It's not like the gene. You handed all the cards. So we monitor it. So when people come in for follow-up, or if we are in touch with them virtually, we note any changes in the household, and it can be divorce, death, move, and the zip code, and put it together with any other traumatic event that have occurred to them, and put it together with the most recent sensor. So we have data. The original sample was 2010. The US is doing it every 10 years. We have data on 2020. So we are updating it now overall and for each individual. We haven't published it yet, but that's what we are doing in the longitudinal study. And this way we will see what about those who stayed in the same environment and those the environment changes. Yeah, because I mean, it's a mobile society. And the virtual helps us track people who are out of state. So we can see those who remained in state and why, and those who have moved on. You know, a good portion of the people are college graduates. It's a network. It's from the greater Philadelphia area. So it's from New Jersey, Delaware. So we follow them virtually across the United States. So we'll be able to see what, it's an important question. We haven't done it yet, working on it.
Hi, thank you. I'm curious about.
I can't hear you. I can tell you now that I can't hear you.
Thank you. So I'm just curious about possibly like a level of, I don't know how to say this, other than profiling for the individual. So if they're young and they're screened and they're seen to be in the PS category, and then maybe they're labeled as that.
Can you come closer? It's harder for me. I'm sorry. But
Sure,
if you come
yeah,
closer,
yeah.
I want to hear you. I can't. But
Okay, thank you. I'm just curious about like labeling individuals. So let's say a young person is screened early and they're labeled as PS. And then because maybe their parents see that or the schools see that, then they're sort of categorized. And then that individual who might have had the resilience to go on a normal trajectory without that label, if they're, because of that label, then they get stuck in that category. So how do you see dealing with
Okay.
that?
A great question. And I'm sorry I didn't mention it. We ask people when they sign the consent for permission to health records and to recontact them in the future. All of them agree. Really, there's no one that said no. So it's some people don't want to do research, but those who enroll would like to. We also ask them, there's a box, do you want to get feedback? Some don't want feedback. And some of them say yes. And some who didn't want feedback, as the kid is growing, remember that they were in a study. We tell them, in the future, we'd like to follow you. You gave us permission. If we could be of any help to you. So people whose kid had a problem and had an MRI, for example, will call and say, I remember that my son, daughter had an MRI. They were involved in a motor vehicle accident and they're doing okay. And they're repeating the MRI and they want to know. So we will send them with their first release of information, a disk of the MRI, so they can compare, especially if they have an incidental finding, because we include reading, a neuroradiologist reads the MRI. If they have an incidental finding, I call them and I tell them, me, for the migrant, the other PIs, and tell them, your son, your daughter had an incidental finding, nothing else needs to be done. We want the healthcare provider, and if in the future, we will send you. Now, for people who want feedback, put aside the MRI, it's simpler, but who want feedback on the assessment of psychopathology screen and the cognitive screen, we tell them that it's a research, that it is not as comprehensive and detailed as one-on-one, because we like to do it on a large group of people. So it's a glimpse. We tell them that. If it's somebody, I share with them, like those who have PS, when we put all the information together, we have a weekly conference. And in the conference, we go, the entire team, over the clinical information obtained by the clinical research coordinators. They have a standard, it's a lot of work. They have a standard summary. They put the summary because they come and go. Most of them go to graduate school, some medical school. After 2-3 years, they're done. We follow them. So there's a standard that we establish. That's what you put in, because you will be going and working on your dissertation. Somebody else will take your place. The kids are important, even when they become adults. So it's a standard summary. And we train them, part of onboarding, to do the summary. And faculty and establish senior research associate are trained, and they go over it to make sure that they are credentialed. And it's so when we give feedback and we ask, it comes on the basis of our observations, parents' report, and medical records. It's you pull everything together when we put it. So parents' report, they know already, for example, they don't socialize. We are the one who make appointments for them, for friends. They never initiate. They're very shy with people. They're overwhelmed when they have to perform, when they have to do a school performance or report. The parents know, the parents are concerned. So what we tell them when they want feedback, we say that the thing that you told us about, we have seen it, that they were more, take them a while to, to warm up, they were hesitant, they seemed anxious, exactly what you see. So it's like, it's a confirmation in some ways. And depending on the age, if they're young, we say that childhood, that children, they're going through the process of maturation. They will not be mature in the 20s. It's not like 18 or 17, 16, they drive a car and they're ready to go now. So this time we will follow them. So we will be able to see what changes. And when we see something of concern, we make recommendation for intervention. So if somebody's, for example, depressed, suicidal, we have a physician or a licensed clinical psychologist on call, because the assessment, when they're virtual, they're done in the evening on a weekend. So they know, and we have a link. So they will ask permission. It happens once, twice a week. We take it seriously. So if somebody, and we'll say, and we have a map of all the healthcare centers that can provide, If it's serious, we tell them you come to the emergency room. And
Thank
if it's, you see, so it's like a setup that puts it, it's research, it's ready to support, make a referral, and see them, even if they're not our patients, if they're close to us, we'll say, I'd like you to come in with your mom or your sister or your grandma, and we'll make sure that you're seen.
I appreciate the bit on the aftercare. That helps. Thank you.
So we need to end soon, but I want to ask a question, Raquel. First, it was a breathtaking talk, and your life's work is so inspiring. And I just want to thank you for everything you've done. It's
Thank you,
really,
Richie.
so all the emphasis is really ultimately, I think, as you said many times, focused on early identification. And if we can identify risk early in life, we have the opportunity to potentially intervene. And one of the things that I often talk about is how psychological interventions, for the most part, even those that are considered the most evidence-based interventions, are really not designed based on what we know about the brain, for the most part. You have an opportunity with all the information that you've acquired and your wisdom over the years to really, I think differently about how early intervention might proceed with kids who are at risk, particularly given what you know about the vulnerabilities in brain function that may predispose them. And as you said a few times, to tweak the trajectory. And, you know, exactly in what ways can you develop a curriculum, a brain-based or neurally informed curriculum to tweak the developmental trajectories? And I wonder if you've thought about that. Because you know, there are the working memory findings. You mentioned self-regulation. There's the increased amygdala activation. There's the work on detecting faces, all of which are, you know, extremely interesting. We know something about the neural networks, and we know that they exhibit plasticity. And so can a curriculum be developed based on those findings that would really be helpful for these kids at risk?
That's a great, I mean, it's a great thought. Very difficult to do. So one of the things that a colleague at Penn, David Mandel, is trying to do is implementation scientist interested in many things, including autism. So he's working with some support of donors and the community is interested in autism to have a school on campus with faculty from the School of Education who are trained teachers and mental health professionals to address, he doesn't think brain, but it's brain, of how to approach from a young age, small, it's like a small school of individuals with autism. So there's a big pen chop initiative in autism of trying to do that. Some of the people from the Autism Center are more brain related. So this will be for one population. So I think it's doable, but it needs a support of everybody. It's like multiple. The state will buy in. It's a big effort. I can think about it more for self-regulation, for example, that cuts across, and it's even in adulthood. It requires an implementation initiative. So that's something that we've begun to think about. We are not implementation scientists. But there are people at Penn, like David Mandel, they have a big implementation, and the Wharton School, people are interested in implementation of what can be done. It will need a sort of like it will be tough, and how to address it in the community. So I've not, I mean, the ASD people have gone, and I've gone to some of the meetings further, but in thinking about it more for the 22Q, how to develop something for them that the teachers can get because they're not familiar, and many health professionals, because it's rare, have not done it. So there it's smaller, but it's more achievable, it's more focused than all of psychopathology. So for example, there, because the transition to adolescence and young adulthood is a major challenge for the parents. When they're little, you know, it's easier to arrange things for them. They grow up and they become teenagers. It's much harder. They're less accepted, they're more picked upon. It's hard for the parents to manage them. So we have begun a parent group to see what are the common, and it's virtual. They participated in the research, but the kids are age 12 to 17, for example. So we listen to, it's a curriculum of education, similar to what we do with psychosis risk and early psychosis, of a little bit more about the disorder, how to advocate. So we tell them how to go to the Office of Disability. I mean, practical things. How to advocate for the kids in school, for special education. We're doing it there. There we are doing it. And how to advocate to mental health professionals who have never seen a kid like that who is different. So we become a resource. They can call us. We have an open house. Invite them. Things like that. That's where Ed participates too, for example. Open their eyes to what's happening. But that it's easier in a focus disorder. But to talk about all of it will need, wow, it will need people with specialty in anxiety, depression, ADHD. So we are doing it for psychosis and for 22Q. But it's something we should think about, of how to reach, you know, educate. I mean, I'll be happy to think to get a group of people, of investigators you think about, and then advocate for it at the, you know, American meeting of the, I've gone to that years ago, of educators of high school or lower school, and prepare something. So they don't know about this stuff. They don't learn about it. So
Thank you.
I think let's do something like that and try. And if we win in Pennsylvania and Wisconsin,
Two
No.
key states.
Keith,
Okay, so Amber, can you give us, there you are. Thank you. Thank you so much, Raquel.
thank you. Thank you.